Dr. Sayir, thank you for your very kind words. I'd like to thank the organizing committee for the invitation. So the history of cancer immunotherapy is a fascinating story, and if we go back 150 years, we will see that Rudolf Virchow suggested that tumors have infiltrates of a variety of cells, and he suggested that cancer develops in sites of chronic inflammation. Now, his hypothesis holds validity even today, because we well know that infiltrating cells in tumors, if you unleash their potential, they can actually kill tumor cells. So the field of cancer immunotherapy has exploded over the past 10 years, and we demonstrate now that we can have personalized therapies, as depicted in this nice cover from the Science Journal, which shows a patient who's receiving CAR T therapy, which is personalized, and you can see that the infusion bag has the contour of his face, just to emphasize how personalized this therapy can be. Now here, this slide, you can see the milestones in cancer immunotherapy. I'd like to emphasize that the adoptive cell therapy concept evolved since the 1980s. The first clinical trial of CAR T cells was in ovarian cancer back in 1996 with disappointing results. The first clinical trial of genetically engineered T-cell receptor was in melanoma. And in 2011, 2014, we had approval of drugs that are immune checkpoint inhibitors, and we reached the point in 2017 where we have the first approval of CAR T-cell therapy for ALL and diffuse large B-cell lymphoma. Now, adoptive cell transfer, as I mentioned previously, the concept existed since the 1980s. And if we think about T-cell therapies without receptor engineering, I will refer you to a trial from the NCI and Dr. Rosenberg, who actually isolated these infiltrating cells that Virchow had thought about 150 years ago. And what he did is he isolated these tumor-infiltrating cells, he expanded them in interleukin-2, and reinfused them into these patients, and these patients had significant responses. These are patients who had relapsed refractory melanoma. And in allogeneic stem cell transplantation, we all know about donor lymphocyte infusions. This was demonstrated back in the 1990 in a landmark paper, which showed that in patients with chronic myeloid leukemia who have relapsed after allotransplantation, if you infuse donor lymphocytes from the donor to the patient, you not only achieve remission, but also cure of this disease. Now, when we talk about T-cell therapies with genetic engineering, there are two types. We have either T-cell receptor engineering or chimeric antigen receptor. T-cell receptor engineering involves, as a prerequisite for antigen processed and presented by the MHC system to a HLA-compatible molecule on the tumor cell, but chimeric antigen receptor does not require that. The other important difference between T-cell receptor engineering and chimeric antigen receptor is that the T-cell, genetically engineered T-cell receptor targets intracellular proteins, while chimeric antigen receptor targets proteins or glycans on the surface of the target cell. And these cells are expanded with interleukin-2, and a prerequisite when you're giving these cells to patients is that you need to precondition with immunosuppressive chemotherapy. Now, the principle of CAR T-cell design is that the chimeric antigen receptor has an intracellular domain, which is usually the zeta chain of the CD3 complex, and it has an extracellular component, which is an immunoglobulin-derived single-chain fragment of an immunoglobulin. Now, how do you engineer a CAR T cell? You take the CAR DNA, you insert it into a viral vector, which is usually a retrovirus or lentivirus, and this is transduced into the T cell randomly. I must emphasize that this is done randomly. It replicates, it's built into the genome, it's translated into a CAR protein and expressed on the surface of the T cell. I must emphasize also here that these retroviruses are modified. They insert the transgene into the T cell, but these retrovirus or lentivirus cannot replicate. This is very important. Now, the first generation CAR T cells involved, as you can see, the zeta chain of the CD3 complex and the construct, the extracellular construct, which involves as I said, the immunoglobulin, a single chain fragment variant, which is specified to target the antigen we want to target. Now, the first generation CAR T cells did not work properly. The problem is that they could not expand, so the signaling into the T cell was very weak. Second generation CAR T cells involve the addition of a co-stimulatory molecule into the intracellular domain, such as CD28 or 4-1BB. Third generation combines both of these, and now we have fourth generation CAR T cells in which we have a protein, a gene expressing a protein is transduced into the CAR T, into the T cell. For example, you have a kind of expression of interleukin 12 combined with the CD28 and 4-1BB. These are fourth generation CAR T cells, and they are now being investigated in solid tumors. So as you can understand, CAR T cells, when they infuse and interact with the antigen target, these cells expand. And one could say that these are what we call living drugs. And with each encounter to the antigen, these cells continue to expand, to replicate. So this is where the CAR T cell, the concept of CAR T cell is based on. It's a living drug. This is a brief history of CAR T cell therapy. First CAR T cell was developed by Zeliger Eschar. In 1996, as I mentioned, there was the first CAR T cell clinical trial in ovarian cancer with disappointing results. In 2010-11, there was a phase 1 clinical trial with CAR T cells in CLL. This was done at the University of Pennsylvania. They had encouraging results, but they ran out of funding, and it was difficult for them to prove that this treatment would be superior to many other agents that were available in CLL. I think the most important landmark in CAR T cell therapy was the first pediatric patient, this young lady you can see in the picture here, who had relapsed refractory acute lymphoblastic leukemia. She was the first patient to receive this therapy, and she almost died. She went to ICU with serious side effects of the treatment, and she managed to survive. And this was a very important turn point in these trials, because it was believed that if she had passed away, this field would have frozen again and probably not expanded as it has now. So it's very important to emphasize that the FDA approved the first CAR T cell therapy for ALL in 2017. A second CAR T cell therapy was approved for diffuse large B-cell lymphoma again in 2017. And just recently, a month ago, the EMA in Europe approved these CAR T cell therapies for use in the European Union. Now, the manufacturing of CAR T cell therapies is a difficult feast, especially if you want to expand this from a lab to a multi-institutional level, and also into, I would say, industrial level, transferring these cells from centers to centers, which are at a very distance. So it's a quite laboring process. It needs strict good manufacturing practice, and the FDA gave a hard time to these companies to get approval. Now, regarding lymphoid malignancies, CD19 is a target antigen that's been extensively studied with CAR T cell therapies. It's uniformly expressed in high density in B cells, and it's important because it has a limited on-tumor, off-target interaction. So it doesn't target other tissues. It's very important. There are a variety of other antigens that have been tested in myeloma and in some other diseases. I will get back to them during my talk. I want to emphasize here that patients with relapsed refractory diffuse large B-cell lymphoma have a very poor outcome. This retrospective study, the SCHOLAR-1 study, demonstrated that patients who have no response to 4 cycles of initial treatment or to 2 cycles of salvage therapy, or who relapse within 12 months after auto, have a very poor prognosis. The overall response rate of these patients to treatment is 26%, CR rate is only 6%, and the median overall survival is 6 months. So the studies I'm going to present to you, the 3 major studies with CAR T cells in diffuse large B-cell lymphoma, involve these type of patients. The first important study is the ZUMA-1, which investigated the activity of axi-cel, which is sponsored by Kite and Gilead. These patients were mainly diffuse large B-cell lymphomas. There were a few transformed and primary mediastinal B-cell lymphomas. I would like to go down to the efficacy. What's very important is, as I didn't mention earlier, the vein-to-vein interval. That means by the time from the time the cells are collected from the patient, sent to the lab to be converted into CAR T cells, and then expanded and taken back to the center where the patient isn't reinfused, this interval is usually around 2 weeks. So the median vein-to-vein interval in this study was 17 days. The overall response rate was 82% with a CR rate of 58%, and there were ongoing responses when the study was published in the New England Journal of Medicine with a CR rate of 40%. The median duration of response was 11 months. Regarding safety, cytokine release syndrome, which is one of the major side effects of CAR T cell therapy, was evident in grade 3 or more in 13% of patients. Neurotoxicity, grade 3 or more in 28% of patients. And grade 5 adverse events in 3% of patients. There were 2 deaths from HLH and CRS, cytokine release syndrome. Here you can see the duration of response curves. As you can see here, those who had achieved complete remission, the median duration had not been reached, and the median duration of objective response was 11 months, while those who achieved partial response, the median duration was only 1.9 months. And here's the median overall survival. When the study was published, it was not reached at the point when it was published. The second study is the JULIET study, which investigated CTLO-019. This is sponsored by Novartis, and I would like to go to the efficacy. The efficacy was 53% with a CR rate of 40%. Median duration of response and overall survival were not reached. Regarding safety, there were no grade 5 adverse events, so no patient died from this treatment. And when it was published, there was an update at ASH 2017, but when it was published, this included only 14 patients with diffuse large B-cell lymphoma. Here you can see the progression-free survival of the patients and the median overall survival of the 14 patients in the publication. It was 22 months. The third CAR T cell therapy that has been investigated, that was presented at ASH last year, is the TRANSEND study, where it involves the CAR T cell, liso-cel, and it's sponsored by Juno and Celgene. I would like to go to the dosage. It had fixed doses. It had a dose level 1 of 5 times 10 to the 7th CAR T cells, or dose level 2 of 1 times 10 to the 8 CAR T cells. The efficacy, the overall response rate was 84%, CR rate 61%. The median overall survival was not reached. But I would like to emphasize here the safety of this CAR T cell. As you can see here, grade 3 or more CRS was only 1%. Grade 3 or 4 neurotoxicity was only 14%. And grade 5 was 2% with no treatment-related deaths. So as you can see, compared to the previous studies, there is a striking difference in terms of safety with less side effects with this CAR T cell therapy. One thing I should emphasize about this study is they actually administered the CAR T cells as an outpatient in a significant number of patients. And it's been demonstrated that it's feasible to give these CAR T cells on an outpatient basis. The majority of patients at some point were admitted to the hospital, but the median duration of hospitalization when it was given as an outpatient was only 5 days. So it's feasible to give these cells as an outpatient procedure. Now, regarding the efficacy, comparing dose level 1 and dose level 2, the higher in the dose level 2, the overall response rate was higher, as you can see. It was 81%, and the CR rate compared to dose level 1 was reached 63% in dose level 2. Here you can see the survival rates. Excellent results in patients who achieved CR, although we need longer follow-up to assess the long-term efficacy of this treatment in this setting. I would like to also emphasize that this is a very interesting case report from the New England Journal of Medicine that demonstrated that CAR T cells also have activity in patients who have relapsed refractory disease and with relapse in the CNS. And this patient has an ongoing response after 12 months, and it demonstrates that the CAR T cells can cross the blood-brain barrier, and it can also be detected, as illustrated in this case report. These cells can be detected in the CSF. Now, shifting to myeloma, we all know that patients who are double refractory to bortezomib and IMiDs have a poor prognosis, and as you can see here, the median event-free survival is only 5 months, and the median overall survival is 9 months. And the initial studies with CAR T cells in myeloma were quite disappointing, especially when using the CD19 antigen, which is usually not expressed on myeloma cells, as we know. But there were CAR T cells constructed for targeting CD138, coupled light chain, which had modest results. But the interest in CAR T cells in myeloma flared up when it was found that the B-cell maturation antigen, which is expressed universally on myeloma cells, is a very active target for CAR T cells. And there are currently 5 big studies running worldwide targeting the BCMA in myeloma. I will just present one study. This was an anticipated study that was presented at ASCO this year, and it's sponsored by Bluebird, Celgene, and the NCI. This had a 3+3 escalating dosage schedule. And if we just go to the efficacy, the overall response rate was around 90%. Many patients achieved what we call biochemical CR, and the safety was very good with minimal toxicity. Clearly, as you can see here in this slide, the response to the CAR T cell is dose dependent. Those who received a dose of 50 times 10 to the 6 had very modest responses. But those who received, as you can see, more than 150 times 10 to the 6 cells had a very, very high response rate. As you can see, the overall response rate in these patients was 95%, complete remission 50%, and very good partial response 36%. So the vast majority had a very good response. And MRD negativity was achieved in 16 of the 16 patients who were studied and had at least achieved biochemical stringent CR at one or more time points of the disease. Now, if we compare the non-active dose, that is 50 times 10 to the 6, as to that of 150 up to 800, as you can see, the median progression-free survival was 12 months compared to 2.7 months. And if we look at patients who achieved MRD negativity, the median progression-free survival was 17 months. Now, if we want to criticize this study, we can say that it does achieve deep responses, and those who achieve early MRD negativity have a median progression-free survival of 17 months. The response is dose-dependent, and preliminary results tell us that the response is independent of BCMA expression, but as you can see, patients do relapse. So the MRD negativity doesn't truly correspond to what we call deep remission in these patients, and of course, it's not yet a curative treatment. So the question is, should we be treating these patients with these cells in a less refractory setting, consolidate post-auto? These are trials of the future. Now, regarding T-cell therapy for relapsed refractory lymphoma, very limited results have been published. What I can say from is that the treatment does work in these patients. You have durable responses. As you can see here, patients are alive at 28 months post CAR T-cell therapy, but it's too early to comment. In CAR T-cell therapies in relapsed refractory CLL, previously we knew that the overall response rate was 50% with, let's say, 25% CR rates. The problem was the hostile microenvironment in CLL prevents the expansion of CAR T-cells. What's been demonstrated recently is that if you combine ibrutinib with CAR T-cells, you can achieve MRD negativity rates that reach 60 and 70%. I will skip the next comment because we don't have time. CAR T-cell toxicities. Patients in the initial phase 1 studies could end up in ICU. And when they first gave CAR T-cells, they wanted a bed available in the ICU in case the patient needed to go to ICU. And there are a variety of cytokines released when you administer CAR T-cells. These cytokines, as you can see here, are detected 36 hours after the infusion. These are the major toxicities with CAR T-cells: cytokine release syndrome, CAR T-cell related encephalopathy syndrome, and hemophagocytic lymphohistiocytosis, which is rare. You can see it in 1% of patients. What we should have in mind is that the more severe the cytokine release syndrome is, the better the response we see in the patient. So if a patient gets this syndrome, it means he's responding. If he doesn't get it, it means he's not responding to treatment. We don't have time, so I'll just comment on the mechanisms of tumor escape. Usually, natural T cells require low antigen density to function, but CAR T cells require high antigen density, as you can see, and it's likely that monospecific CAR T cells will be unlikely to eradicate disease in this setting. And the other thing is that you have T-cell exhaustion, you have excessive T-cell receptor zeta chain phosphorylation, which causes anergy of these cells. How much does this therapy cost? We all know it costs around $500,000. Yescarta, which was approved for diffuse large B-cell lymphoma, the price is a little lower. Liso-cel, which will probably get approval because it is effective, but also it has minimal toxicity, and it can be probably administered at an outpatient setting. The price tag has not been yet mentioned, but it's probably going to get approval. So it is a very costly therapy. I'll just remind you something I bumped into a few days ago on the internet, that the NICE committee in the UK rejected CAR T cell therapy for the NHS. It refuses to pay this price for the treatment of CAR T cells. Regarding the CAR T cell therapies studies, although worldwide North America and Europe dominate clinical trials, as you can see in CAR T cell trials worldwide, China is dominating the field with North America, and as you can see, Europe is not contributing to this field in science. Future of CAR T cell therapies, this is the last slide. New targets will be apparently targeted in the future. What's very promising is gene editing, because the construct is inserted randomly into the T cells, but when you use gene editing, you can actually target where you want to insert the CAR. This is very important. We don't have time to discuss it. Smart CARs, where you can actually regulate and switch off the CAR T cells if you want to. In the future, we might be seeing chimeric co-stimulatory receptors. So you might see PD-1 co-stimulatory molecule with CD28. Bispecific, you might have a combination of CARs expressed on the same T cell. And the last comment is that in the future, we might even see off-the-shelf CAR T engineered cells, which if we can overcome the immune barrier and produce universal off-the-shelf CAR T cells, this will be a very important landmark in CAR T cell therapy. Thank you very much.